I'm going to go ahead and invite you and John up for the panel discussion since we've run over just a little bit, and that way we can have both the questions for your talk and for the panel in general right now. So we have one question at the microphone already. Please identify yourself.
I'm Risha Putzrath with the Navy and Marine Corps Public Health Center, and I'd like to take your integration of genetics and epigenetics one step further in the regulatory context. As I'm sure you're aware, a lot of the relative potency of both PAHs and other carcinogens that use the AH receptor is established on the basis of effects such as AH binding or other in vitro tests of that nature. And so I would like, if you would be willing for you to speculate on the implications of your analysis on translation of issues such as relative potency and also high dose to low dose issues, if you would.
Absolutely. Of course, you managed to ask 5 questions in one.
Pick your 2 favorites.
Only one question.
So what I'll do is I'll try to dissect it from the bottom up, since that's the way that I probably will remember it. The idea of low dose versus high dose, I think probably we're going to have to rethink those definitions a lot. And of course, that's not surprising to you, since I know that we've all been moving towards looking at lower doses. The major conceptual shift that I think needs to take place is that when we think of chemico-biological interactions, we normally think of outcomes which are way down in the progression towards injury or disease or negative outcome. We're going to have to start rethinking that, because we now don't need to really worry about dangers that will kill us right away, right? The real question is, what are environmentally relevant doses of agents which regulate AH receptor doing to dysregulate the machinery of cells? And I think data such as this basically tell you that number one, you're going to start seeing effects that are a lot more discrete, more subtle, and that perhaps the consequences of them have to be sort of accumulated over time before you're going to see them. The other question that you asked, I think, related to the screening, machinery and tests that we have in place right now. I think that natural evolution will probably take care of that. We will continue to monitor good assays out there, that we can implement to continue the screening process and hopefully minimize the depth of analysis that will be required for some of these agents.
Yeah, Jim.
I really like both of these talks. This idea of the low dose setting you into sort of a new biological state, of taking this plasticity and moving it to a new state, reminds me a lot of the whole problem we have with antibiotics. We can give them, kill the organism. It's great for our patients. But if we give too low doses, we select it. We give it enough time for that rheostat to evolutionary change, make a molecular change that makes it be able to survive, basically evolve under these conditions. So I actually worry a lot about these low-dose things because they reset the evolutionary paradigm.
Absolutely.
So I had a follow-up question to that, actually. And so first of all, Ken, I think your idea that there's the reprogramming of the network is really exciting, and I think that's going to turn out to be very important. The question I have for both you and John is, experimentally, these are the tools that we have, but you're really looking at an L1 storm when you're infecting these cells. You're not looking at the rare event where one L1 in one cell moves to another place. So how much of what you're seeing, and perhaps even some of the things that John's working on, might be a reflection of the storm and may or may not actually be happening in terms of the individual cell in which the jumping is occurring?
So, yeah, I like that L1 storm. So I guess for rewiring networks, there's two things to consider. So we're heavily biased, as you said. We just want to make something jump and apply engineering principles to then figure out why it works. And that's basically what we've been doing. But the idea of creating networks, this is, I love Ken's data, and it's also been an idea, at least in a theoretical sense, that's been around since the late 60s. So when Orgel and Crick and others were basically, Doolittle and Sapienza were saying that studying junk DNA was intellectually futile as to why it was there. Roy Britton and Eric Davidson put out a very provocative paper that said that as L1s or any repeated DNA is jumping about the genome, that there's a way to distribute DNA sequences to new locations. And then after they get there, these sequences could fall under Darwinian selection. So imagine a situation where you had a transacting factor that would bind a cis-acting site, and that cis-acting site happens to be in a mobile element that's been distributed about the genome in many, many copies. Now you have the possibility where certain elements at given genomic locations, that pieces within those elements could fall under selection and then rewire networks. And I think there's evidence that's coming up for this now. I would caution that that's not why L1s are there. That just simply happens to be the result of a by-chance evolutionary process by which you distribute these elements around and then the host figures out a way to use them. So in that way, I think that's it. As far as, I totally accept the L1 storm and what we're doing, and then how we extrapolate results gained from a transient overexpression assay where we want L1 to jump into normal regulation. That's a challenge any in vitro biochemist geneticist has to deal with. And then the ways to do that is actually then to progress and try reductionist experiments. So if I overexpress a protein I find that interacts with L1, are there consequences? Can I move it in vivo? Perhaps. People like Scott Devine have done beautiful work right now where they're finding L1 insertions in cancers, certain types of cancers, by very good methods. And now the question becomes, are they driver mutations? Are they not? And the question is, are there L1 storms in vivo? So could there be certain disease states where actually the de-repression of global methylation now provides a way that you could transcribe these elements, and if you're laying a lot of eggs in the form of ribonuclear protein particles, might that provide a permissive environment that you actually have high hoppers? And can those high hoppers then create mutations? I think these will be beautiful areas to study, because if that's true, then there'll clearly be an environmental and epigenetic intersection, as Kenneth referred to.
I agree with what John said. Just remind you and emphasize the point that he made, that there in fact could be L1 storms out there. Point well taken, though, that it's an artificial system.
Yeah, David.
Yeah, Dave Gilley, Indiana University School of Medicine. The question I have is, we're learning all these interesting things about from the ENCODE project about the retrotransposon elements influencing transcriptional control. Are there, and you might, I apologize if either one of you have already mentioned this, but are there examples in humans of transposition driving normal, healthy development in some way? Or are these disease states?
Actually, we probably both forgot to mention the fact that the time during human development when L1s are most active is actually during early embryogenesis. In fact, during early embryogenesis, up until probably the 8-cell stage, what you've got during that shuffling of genetic material is haywire L1 activity. So that much, I think, which I think in some ways I kind of think this is a debatable point, what L1s are meant to do. I think in many ways, whatever the outcome of that genome is going to be a function of that reshuffling that's taking place.
Right, so to Specific examples, yes. Mouse thymidylate synthetase gene, its polyadenylation signal is derived from L1 sequences. There are growing examples as people start to look that there are cis-acting regulatory elements that are there for gene expression that are derived from transposable elements. These are just going to start cascading in. For transposition per se, no. So what like that? It's normally just. What was the question exactly?
The question is, do you actually see jumping in transposition events that are involved with a developmental process, a healthy, normal?
So what Rusty Gage has seen, he's actually made transgenic mice having a human L1 tagged with one of the GFP beacons, and one of the places where he and Alison Muotri have detected retrotransposition is in the brain, in the basically in renewing cells in the brain, stem cells, as they're basically moving through a differentiation process. Jeff Bailey's followed that work up using deep sequencing techniques, and he's got evidence right now that there are endogenous retrotransposition events that he could detect there. The question with those processes are validations. So you get big numbers, and then the validations funnel down to relatively few. So there are instances there where people, at least through a variety of studies, and we've actually derived neural progenitor cells from human embryonic stem cells and shown they're quite receptive for retrotransposition in vitro with our engineered L1 vectors. Jose Luis Garcia Perez has seen that. Whereas in other cells, you really don't see it. Another case where it actually happens normal developmentally, one would argue, is the first somatic insertion that was picked up in the added APC gene. It's got a long name. So basically the thought is it's a single hit that led ultimately to colon cancer, and then when that was looked at by Miki et al., they haven't been able to find that hit in the surrounding constitutional tissue. So that gave the first idea that you presumably got an L1 hit, maybe in a stem cell, that basically led to an activation of one tumor suppressor copy, you twist the other one by LOH, and then out a tumor goes. And that set the stage for beautiful work by Scott Devine, where he showed L1 jumping in certain types of lung tumors. More importantly, what he actually showed in those cases was that the cells that actually received the new retrotransposition events tended to have a hypomethylated genome as compared to the ones that had no retrotransposition event. So again, that's a tantalizing clue, though a small study. I think it's an incredibly important study by Iskow et al. That says that there could be a link between the epigenetic state of L1s at a given locus and their ability to jump. So there's a lot of anecdotal reports out there. Now, does this mean that L1's important for that? I guess that's the point of arguing, because it gets back to this chicken and egg argument. Is it just the result of a resultant phenotype, if you will, of a cell, or does it have a purpose? And I come from the junk DNA world, and I've started to be okay with the idea that these are not purposeful, but they're impactful. And impactful, too, is a lot of ways just by doubling yourself and setting up new templates for Darwinian evolution. That's a way that, you add more sequence, just use it. That's kind of what we do in biology.
Okay, so we have, I think, 3 or 4 speakers and 5 minutes left. So I'm going to ask for very short questions and short answers.
Yeah, that's not my strong point.
Neither is mine.
You had mentioned that HDAC inhibitors as a potential therapeutic. So the very first HDAC inhibitor comes off of patent this year. That's how many years they've been around. And we don't, in the clinic, understand the mechanism of how they work. Do you think that what you've offered, is in part an explanation of its mechanism?
I wouldn't venture to go that far.
Good short answer. Okay, next question.
Great talk, Ken. So it's actually for you. So I was intrigued by the idea that oxidative stress was, when you're treated with benzpyrene, it's the oxidative stress that's inducing it. So do you think that chronic diseases like irritable bowel, that you're actually getting a line storm in that situation, and that's what's causing the dedifferentiation of the tissue?
I actually think that there's some merit, to that idea, because when you look at the cancers which have been associated with L1, de novo insertions or L1 storms, if you would, typically those are cancers where the environment is a highly inflammatory environment. Data which I did not show is that if you were to blow up the regulatory network that I showed you there, much of what blows up is all inflammatory related genes, a whole bunch of cytokines, and a whole bunch of virally regulated antigenicity receptors, which I think open up the question in terms of what is the intersection between inflammation and L1 regulation. And we've wanted to do those experiments but haven't gotten that far.
So just an interesting analogy there. Those same inflammatory environments drive changes in DNA methylation, however, in the opposite direction, toward hypermethylation, not hypo. Yeah.
Absolutely.
Yeah. John, did you want to say something?
No, clearly L1's inflamed our genome.
That was better than I did.
No.
I just have one little comment, if I can keep from pushing. I've heard Darwinian selection, Darwinian evolution come up a couple of times. I just want to reiterate that we humans aren't set up well for that strong positive selection, just given our population genetics, but we are set up to accrue mildly deleterious mutations.
Sure. So the idea too is you got to remember that these elements go all the way back to the start of mammals, and that many of the situations where you've actually found these cis-acting sequences that have been co-opted for gene expression purposes, they're not human inventions, and so they go all the way back. And at the deepest end, Tim Bester is the first to champion this argument. You could consider L1s as sexual parasites that simply are jumping about the data genome. And Donald Hickey has done some beautiful population-based analyses. Again, this was first pointed out by Tim Bester or resurrected by him, that L1, as long as even if it's mildly deleterious or somewhat deleterious, as long as it doesn't just toast the organism, it'll drive to fixation anyway. And this is one of the reasons to start to say that L1 is basically doubling at twice the rate of any other gene in the genome. It's going to take over as a sexual parasite. That's probably why we have so many, which is why I'm reluctant to say they're there for a purpose, because the net outcome of something that spreads through sex and jumps around in that way is to get more. And last thing just to bring up very quickly is that genes that knock out DNA methylation, like DNMT3L, people have shown that L1 expression goes way up. Now the question is, do you get jumps, and could you have a storm there? And I think second-generation sequencing would be a way to start to sort that out.
All right, last question.
Okay, it's not a question, it's a quick comment. There are several human disease conditions, for example, childhood asthma, and also in elderly, when they're exposed to like polluted air or even transplacental PAH exposures, they do have line 1 hypomethylation. So whereas if you compare in the same group of individuals, they might have different either hypo or hypermethylation of various genes. So it does normally in these population studies, they just viewed that this line 1 hypomethylation as a sort of like reporter for global hypermethylation, and kind of not pay much attention to what the consequences might be. So I think it will be important that for the scientific communities to start paying attention to it and not just viewing it as some general reporting mechanism.
So yeah, but there's two things there. One is I do think it is a general reporter of somatic cells, et cetera, and it's very useful that way because of their interspersed nature. Two is that the methylation also takes on a host defense challenge. In fact, in the DNMT3L example that I mentioned, Bester's shown beautifully that this de-represses L1s at certain stages during male meiosis, and that could actually lead, the de-repression correlates with male meiotic catastrophe. So the thought there is there a direct connection between L1 activity and jumping and the failure of spermiogenesis that actually occurs in those animals. So this is, there are clearly places where one could make very strong arguments that the goal in germ cells is to keep the transposable elements in check. Then when it comes out to actually looking at, say, cancer cell A versus cancer cell B, L1s are a very useful tool to basically give you an idea of, like, what's the genome environment look like with respect to methylation. So I think the two are separable in that respect.
Now,
Closing comment, Ken?
The only thing I would add to what John said, which I agree with, is the idea that remember that not all methylations are created equal, and that what we need to be distinguishing is methylation events which are impactful to reactivation of the element versus generalized, methylation events that may be of no real biological consequence. And then the readout versus the functionality might actually be a function of those two differences.
All right, fantastic. Well, I would like to thank again both speakers. They were fantastic talks. It was a great discussion. And just, we're going to take a 10-minute break. We're going to come back at 10:45. Those of you who are on the web, please remember we're going to be having a discussion of implications and research needs. Send those in, and we will take your questions and comments in our panel discussion that we're going to have at 10:45. Thank you again.